Hello, everyone. Welcome to the Roche Sequencing webinar series, where today we'll be talking about a new target enrichment technology that Roche has recently launched called KAPA HyperPETE. And so with that, I would like to give you a high-level overview of what this novel target enrichment capture method is all about. Well, first and foremost, like most target enrichment technologies, it's going to use shotgun libraries prepared from DNA or RNA as an input into capture. Next, you may be asking yourself, well, what does PETE stand for? Well, it's an acronym that stands for Primer Extension Target Enrichment, therefore KAPA HyperPETE. So what advantages does this give you? Well, what it does, and first and foremost, most importantly, combines hybrid capture performance with the ease of use and turnaround time of amplicon workflows. In addition, it offers an automatable single-day workflow. Next, detection of all major variant classes. This includes single nucleotide variants and polymorphisms, short insertions and deletions, copy number variants, microsatellite instability, or also known as MSI, and last but not least, fusion transcripts, known and novel. And with that, I will turn it over to Dustin Masser.
All right, thanks, John. So now we'll go into how does the HyperPETE technology work. So as we described previously, the input into the HyperPETE capture is a shotgun library. Now, I won't go through the making of a shotgun library, but you can see that we have our library molecules with our adapter sequences ligated on here. We do use our KAPA unique dual index adapters, and you have the option of using these adapters with or without unique molecular identifiers. Now, for somatic mutation calling applications, we will use UMIs. So in this example, we have our input shotgun library, and we have on the top our target molecule in blue, so the insert here. And this one is gene A exon 10, for example. Now, all the other targets or library molecules within the sample themselves are considered off target here, and those are represented by the other colors. The initial part of the HyperPETE technology uses a capture primer, a target-specific primer that is biotinylated, and that anneals to your target of interest. Second, it performs an extension product, so a single cycle of PCR through the rest of the library molecule, essentially performing an extension reaction and generating a probe. Next, we use streptavidin beads that bind to the biotin labeled extension product that has captured the library in a capture step. Following the capture, we do wash away off-target molecules. Therefore, all that's left are our on-target molecules initially that have had that capture extension reaction performed. A unique part of KAPA HyperPETE is that we do use a second target-specific primer that increases the on-target rate of the technology that will bind upstream to the initial extension product. And then this will extend through that initial product and effectively release all of the target molecules into solution. And this allows you then to transfer the supernatant from this reaction into a new tube. Following that transferring into a new tube, we perform a post-capture PCR using the Illumina P5 and P7 amplification primers. And this enriches our sample to be ready for QC, pooling, sequencing, and analysis. Now, as John mentioned, the KAPA HyperPETE technology is meant for a fast turnaround time. So here in this graph on the left, you can see that we have turnaround time in hours for KAPA HyperPETE compared to hybrid capture and other amplicon methods. So right off the bat, given that hybrid capture methods typically use an overnight hybridization workflow, you can see that the KAPA HyperPETE technology is way faster. Now, compared to the other technologies here, like the anchored multiplexed, we have a very similar workflow time. Now, one of the benefits of KAPA HyperPETE compared to typical amplicon methods is that you can process fewer samples than these amplicon technologies when trying to cover long contiguous regions. So looking at the HyperPETE application turnaround time, you can see here on the x-axis we have listed in hours. And we have components of the entire workflow, including library prep, the enrichment, QC, and library pooling. Now, the HyperPETE turnaround time differences are really dependent on the library preparation method. So here we have examples of the somatic tissue DNA workflow, the cell-free DNA workflow, and the somatic tissue RNA workflow. And you can see that there are differences in these workflow times listed here. So the HyperPlus workflow includes enzymatic fragmentation ahead of DNA library preparation. And that's why you can see that it's slightly longer than the KAPA HyperPrep, which does not include fragmentation. And then finally, the KAPA RNA HyperPrep, which includes cDNA synthesis ahead of library preparation, is the longest workflow, being completed in about 5 hours. Now, one thing to note, you can see on the graph here in the darker blue sections next to the library prep sections, is that the enrichment workflow is the same across all applications. So here we're looking at a coverage comparison, looking at HyperPETE versus an anchored multiplex method. And this is, you're looking at an IGV view of a region of the APC gene. So on the top here, we have reads that are aligned from the anchored multiplex workflow. And on the bottom, we have reads that are aligned from the KAPA HyperPETE workflow. So right off the bat, one thing you can notice is that there's superior performance and excellent coverage uniformity across this target region in comparison to the anchored multiplex, where effectively you have lower coverage uniformity across this region because of the amplification and enrichment methods that PCR and anchored multiplex use. So now I'm going to hop into some data, preliminary data for both the somatic tissue DNA workflow and the cell-free DNA workflow, as well as the somatic RNA workflow. So our somatic tissue DNA workflow here, this panel is using the HyperPETE pan-cancer panel. And on the top here, you can see that the gene and target lists for this pan-cancer panel, so the genes are listed here. The panel content includes 1,132 CDS regions across these 86 oncogenes listed, and they cover single nucleotide variants, insertions, deletions, and copy number variants. In addition to this panel content, we have also included 190 MSI loci for characterizing microsatellite instability status of samples, and that's listed in that final box in purple. The capture panel size of this panel is approximately 300 kb. And the samples used in this study were FFPE tissue samples, so 6 low quality, 6 medium quality, and 4 high quality normal adjacent tissue samples. And these were used to assess enrichment metrics. We also included cell line reference samples, so a Sera-seq lung and brain CNV mix, a Horizon structural multiplex reference standard, and genomic DNA representing microsatellite stable status, as well as 2 custom mutant gene mixes for MSI gene DNA mixes. So the FFPE was extracted using the KAPA NGS DNA extraction kit, and the quality was measured using the KAPA NGS FFPE DNA QC kit. These libraries were then generated using our HyperPlus kit in combination with KAPA NGS FFPE DNA polishing kit and the universal UMI adapters. And we used inputs of either 10 nanogram or inputs based on quality assessment. The libraries were captured using the KAPA HyperPETE reagent kit and then sequenced on the NextSeq 550. We subsampled reads to 30 million paired end reads and analyzed using our Navify mutation caller to assess enrichment and variance detection performance. So here on the left, you can see the QC metrics of the tissue DNA representing our enrichment performance. On target rate was 69 to 78% with good performance across all samples and input amounts. Below that, percent panel exon bases greater than 300x and greater than 500x were 95% across all samples when using quality adjusted input. Percent panel exon bases greater than 300x and greater than 500x were variable, but predictable when using strictly 10 nanogram input. Low quality and mid quality DNA resulted in lower performance compared to higher quality DNA. On the right, we have our variant detection performance. And so you can see, I'll start with the SNVs and InDels. 99.2% of these short variants were detected across all reference cell line and cell line mixtures with variant frequencies of approximately 5%. 90% of copy number variants were detected with a single CNV call being low confidence. So we did detect all of these CNVs, however, one was called low confidence. MSI status was correctly assigned for all 12 of the MSI FFPE samples and 6 MSS cell line samples. So 100% of the microsatellite stable calls were called, as well as 100% of the MSI calls. So next I'll show preliminary data for our somatic cell-free DNA workflow. And again, this workflow is using our HyperPETE pan-cancer panel, which I described previously. One thing to note with cell-free DNA as an input, the CNV calling is not enabled. Additionally, MSI is not enabled in our workflow for cell-free DNA samples. The capture panel, again, is approximately 300 kb. And the samples used in this study were 3 healthy donor plasma cell-free DNA samples, again, used to assess enrichment performance metrics, as well as 3 Sera-seq ctDNA complete mutation mix samples with the catalog numbers listed, and reference cell line samples to assess enrichment and variant detection performance. So again, here on the left, we have our QC metrics for the reference DNA. On the top, we have the on-target rate, and this is representing 67 to 74% with good performance across all of the input types. The percent panel exon bases greater than 100x was 85% across all sample types. Better performance was observed with higher input amounts and when using cell-free and using cell line DNA as an input. The percent panel bases, exon bases greater than 300x showed similar trends to the 1000x metric, but with a larger observed impact with lower input. Now, of note, increasing read pairs used in this analysis from 60 million to 110 million, the percent panel exon bases greater than 3000x improved, ranging from 73% to 95% for the 50 nanogram input. Now, this data isn't shown, but it does demonstrate that increasing read depth for this application will lead to better performance for this particular metric. On the right here, we have our variant detection performance. And again, we're only looking at the short variants, so the CNVs and InDels. On the bottom here, we have 100%, showing 100% of short variants at the 1% allele frequency were detected across all reference cell line DNA samples and all input amounts. On the top of this table, we have 88.2% to 94.1% of short variants at the 0.5% allele frequency detected across the reference cell line DNA samples. So the variants here that were not detected, they were present, but they didn't get called due to threshold settings applied in the analysis software. Now, of note, you can change these depending on your research needs. And finally, I'll wrap up with preliminary data for our somatic tissue RNA. So the somatic tissue RNA workflow uses our HyperPETE lung cancer fusion panel. On the top of this table, we have all of the genes included in this lung cancer fusion panel, and below each gene, we have the exon lists included. So this panel content includes 16 oncogenes covering the 137 exons designed to detect both known and novel fusion partners at both the 5' and 3' end of these listed exon junctions. We also include 4 housekeeping genes to serve as positive enrichment controls within the workflow, and those genes are listed here. These genes they are expressed at varying levels across different tissues, but they are locally expressed genes. The samples used in this study are 4 normal adjacent FFPE RNA samples, again, used to assess enrichment performance, as well as 2 cell line reference RNA samples, including this Horizon fusion mix and the Cereseq fusion RNA mix, to assess enrichment and the variant detection performance. Now, the RNA quality was determined using the DV200 score from the Agilent RNA 6000 Pico assay on the bioanalyzer. Libraries were generated using the KAPA RNA HyperPETE kit in combination with the KAPA Universal UMI adapters, using either 10 nanograms or 50 nanograms of RNA while adjusting PCR cycles based on the input amount and the DV200 score. The libraries were captured using the KAPA HyperPETE reagent kit and sequenced on the Illumina NextSeq 550 instrument. These reads were subsampled to 540,000 paired end reads and analyzed using the Navify mutation caller to assess the enrichment metrics and variant detection performance. Again, on the left, we have our QC metrics of the tissue RNA. So following ribosomal RNA read removal, which represented about 2 to 13% of all reads, the on-target rate was 92 to 97%, with good performance across all sample input amounts, qualities, and types. So showing here, regardless of the input, we have very good on-target rates. The housekeeping targets, which are used as enrichment controls, are considered on-target reads and make up between about 40 to 60% of the reads, depending on the sample type, and that data isn't shown here. Again, on the right, we have our variant detection performance, and we're looking at these known fusion variants, looking between 10 nanograms and 50 nanograms. What you can see is that 100% of the fusions in the reference cell line RNA samples were detected. So as a summary, the HyperPETE technology is leveraging Roche's content and extensive design expertise in order to investigate difficult regions of the genome. The workflow allows you to save your precious samples and improve sensitivity with no panel splitting. It provides superior sequencing efficiency with high uniformity, focused panel designs, and no primer trimming. We are going to be offering fast custom panel ordering turnaround time for rapid deployment and testing. And again, the workflow is optimized for somatic variant detection from cell-free DNA, FFPE tissue, and RNA samples.